All right, this is Unit 3, essay video, or essay question number 3. There's 2 parts to this one. I'm just going to do the first part right now. So we're going to describe the composition of blood, include the components of plasma, describe the mechanism by which red blood cells are removed from the circulation, especially explain how heme is broken down and what happens to the various components of heme. So first we'll talk about what makes up blood. So just picture this as a sample of blood. It's split up into 2 parts with this black line. This part up here is the plasma, and that makes up 55% of the blood. And then this bottom part is the other 45%, and that's called the formed element. And we'll start with the plasma. So plasma, again, it's 55%. 92% of that is water, so the majority of it is water. And then within the plasma as well, we have a few different, what do I want to call them? I don't know if they're proteins. And so one of them is albumin. And we talked about albumin in the video about, what was it called? The capillary exchange. Because if you remember, albumin was really important with the capillary exchange because that's what determined the BCOP. And BCOP stands for, I can't remember the B is, but it's colloid osmotic pressure. And so it might be blood, colloid osmotic pressure. Because I think the other one's the ICOP, and that's interstitial colloid osmotic pressure. So this is the colloid that determines BCOP. So if you remember right, if we have high amounts of albumin, it's a big protein, so it can't pass through the membrane. And so if we have high concentration in the plasma compared to the interstitial fluid, that's going to draw on water from the interstitial fluid. And so that's albumin. It makes up about, I think it's said in the book, 60% of these proteins, and so it's definitely the majority. We also have globulin, and that one I think it was talking about, does something about with transport and antibodies. I didn't get much information about it from the book, but that's kind of what it mentioned. And then we have fibrinogen, which we talked about quite a bit. We'll talk about that in blood clotting and stuff. But it's a clotting factor within the blood, and so that's important. And if we have the plasma and we remove the clotting factor from the plasma, I'm pretty sure it's from the plasma, then we're left with the serum. Book mentioned that, don't know how important that is. But then we'll get down to the formed element. That's where we have our cells. And the majority of the cells in the formed element is the red blood cells. It's really pretty much all of it is the red blood cells. And then here in the green, between the green and the black line, that's what we call our buffy coat or buffy layer. And that has white blood cells and platelets. And white blood cells are for immune system response. And then the platelets are also used for blood clotting as well. But the majority has the red blood cells. And the red blood cells, those are made from in the bone marrow. And I can't remember, but there's some crazy numbers with, I'm back. I don't know when that cut it out, but yeah, red blood cells. Like we talked about, it's a huge percent of the blood. Well, it's a huge percent of the formed element, which is a large percent of the blood. And there's something that we can do. It's like a measurement or a blood test, and it's called the hematocrit test, or I don't know if it's the hematocrit test, but it's called the hematocrit. And what that is basically, it's the percentage of the blood that is red blood cells. And I know it's like around 45%. For females, I think it's 35 to 45%, and then for males, it's 45 to 55% on average. And that percentage can change a little bit, because if you're in situations or an environment of low oxygen, your body's going to produce more red blood cells, because the red blood cells are what transfer or I guess are like oxygen carriers. And so the more oxygen carriers you have, the more oxygen you'll be able to deliver to your cells. And so in environments of like low oxygen, like high altitude, or if you're doing a lot of cardio training, your body's going to be used to kind of a more low oxygen environment, or your body's going to be used to having a high demand of oxygen. And so your body's going to produce more red blood cells. So if you're going to have a higher hematocrit or higher percentage of red blood cells in your blood due to those reasons. The reasons why it could be low is there's different types of anemia. Anemia is just like either you have low red blood cells, or the heme, we're going to talk about in a second, or like the hemoglobin that actually carries the oxygen has a problem. But pretty much it's just like your cells aren't getting the sufficient amount of oxygen they need, and that's what anemia is. And there's like iron anemia, like iron deficiency anemia, and that's due to low iron. And iron's important because that's an important factor in making red blood cells. And so yeah, that's probably all I'll mention about that. There's different types of anemia. We'll just talk about that one right now. Yeah, that's what makes up the blood. Pretty simple from what I know, but I know it's a lot more detailed, but just from what I've learned so far, this is kind of what I've covered. But yeah, let's move on to over here. This is, so we talked about, we have the red blood cell. I'm gonna draw it in blue. I'd like to draw it in red, but red markers across the room. But a red blood cell, it's like disc shape. I don't know if this is 100% accurate kind of drawing, but as you can see, it kind of has this concave structure. And this kind of helps with the red blood cell, with its flexibility. The important thing I wanted to bring up is on the red blood cell, you have hemoglobin. And hemoglobin, there's a bunch of them on the red blood cell. I can't remember the number, but it was like a huge amount. But these hemoglobins just kind of attach and hang on to the red blood cells. And the hemoglobin are actually the things that actually carry the oxygen. So the hemoglobins are kind of like piggybacking on the red blood cells as they travel through the blood vessels. So, I'll angle the camera a little bit more. So a hemoglobin, this is like a hemoglobin structure. It has like 4 different parts. They're pretty much all exactly the same. But let's just talk about one of these sections. I drew it, hopefully you can see it, but this red, it's kind of like this chain or rope that's all kind of looped around each other, wrapped around each other. And that is the polypeptide chain, and that's called the globin. And then with that, there's a kind of like a disc, which I drew in black, and that's the heme part. And then inside of the disc, there's the iron. And so you take heme and you take globin, the heme, the disc, the globin, I think it's amino acids, the amino acid chain, or polypeptide chain that's all wrapped up, and you get hemoglobin. And so that's where that word comes from. And then the iron it attaches on there. And yeah, there's, again, for one hemoglobin, there's 4 of these sections, or there's 4 of these sections that I talked about. And so here's all 4 of them. The names of them are the beta chain 1, beta chain 2, alpha chain 1, alpha chain 2. And then something that was mentioned in class is the fetus. They have, instead of having a beta, they have gamma chains. And I'm not really sure when that changes, but the fetus starts out with the gamma and the alpha, but then later on it changes to the alpha and beta. Important with this, like I said, the hemoglobin is actually what carries the oxygen, and you gotta picture this as, this marker's not doing great, but this is one like hemoglobin right here. And usually they're all kind of like, I drew them more separately, but they're all kind of like bunched together, connected. Each hemoglobin can carry 4 oxygens. So pretty much each little individual one with its polypeptide chain, heme group, and the iron, that can carry 1 oxygen. This one can carry 1. This one can carry 1 oxygen. This one can carry 1. That one can carry 1. So we get 4 total. Yeah, that's kind of the breakdown of blood, and then kind of an in-depth look at hemoglobin and how that ties into the red blood cells. Now, we also want to talk about, this might be a little too close, come right here. So now we're going to talk about, like kind of like the recycling of red blood cells. So we talked about red blood cells. They're made in the bone marrow. This is I know you can't see me, I'll angle it a little bit. Hopefully that's good enough. But this is a blood vessel, and in the blood vessel we have a red blood cell. And when the lifespan of a red blood cell is about 120 days. It's made from like a stem cell. There's a chart in the book. I didn't really know how important it was. Didn't really pay much attention to it because it looked a little bit kind of like a lot. But you get a stem cell. From the stem cell, you can get like red blood cells. You can get white blood cells, different stuff too, probably. But that's where the red blood cell comes from, and it gets popped into the bloodstream. And at first, it's popped into the bloodstream as like something else. It's like kind of a baby red blood cell, but then like within a day, or like 24 hours, it matures kind of and becomes a red blood cell, and then it will be in the bloodstream for around 120 days. But then once it starts getting old or it gets damaged, starts to break down, we'll have a, what is the word of the thing that gobbles it up? I can't remember, but I think it's a macrophage, comes, kind of eats up, engulfs the red blood cell, and then it starts to break it apart. And so the macrophage starts to break it apart. It will take the globin group, which was the polypeptide, it will break it down back into like its amino acid chain, and then it'll like send it back to be reused for protein synthesis. I think that's back in the bone marrow, but I think the book said something also, it talked about like the liver and maybe another organ, but pretty much the essence of it is put back, kind of recycled to make new red blood cells again. Also, the heme group, remember there's 4 of those with the heme and the iron. That's broken down into the heme group and then to the iron. And those both can be either sent further on, which we'll talk about in a second, or both of them can be sent back to be like reused for protein synthesis to make red blood cells. So the heme and iron can be sent back to here, back to kind of the recycling station. But then they can keep going. Iron, it can be bound to transferrin in the bloodstream as it kind of travels through. Then finally it will make its way to the liver. And inside of the liver, I think it just kind of gets stored. I don't know if ferritin is like where it gets stored or it's on ferritin and that's how it gets stored in the liver. But ferritin is kind of the player that assists in storing iron in the liver. The heme group, if it doesn't go back to the recycling center to be reused, it's going to take a little bit longer of a journey, and it's going to go to biliverdin. So when it gets to the heme group, as it loses the iron, it becomes biliverdin. And then pretty quickly, from what I understand, I think within like the bloodstream, it gets turned into bilirubin. We didn't go that much in depth in the class or in the book, but it gets turned into bilirubin, and that's kind of what we more focus on. And from bilirubin, it will go from the bloodstream into the liver. And inside of the liver, it's bilirubin, but then it's going to get attached to it these glucuronic acids, and 4 of them will attach to the bilirubin. And once that happens, it gets a name change and it becomes conjugated bilirubin. And then while it's inside of the liver, it's going to then be secreted or transferred into the bile or the the intestine. I'm pretty sure it's a large intestine. But once it gets into the intestines or the bile, I can't remember exactly what it does, but these glucuronic acids get removed and they get taken off, and then it's kind of left with just by itself. But now that's, before we call it bilirubin, but now it's in the bile, we're gonna call it urobilinogen, I think is how you say it. And so right now, it's got its glucuronic acids removed. Now it's urobilinogen, and it can do a couple different things. Urobilinogen in the large intestines, one of the things it can do, it can go back to the liver, be sent back there, and that's called the enterohepatic urobilinogen cycle. So it can just be, kind of like a cycle. You see it's just going to be here, it's going to get its glucuronic acids back into the bile. The glucuronic acids are going to be taken off. Now it's urobilinogen, and it's going to go back to the liver. That's why they call it that cycle. But that's not the only option. Another option, it can go up to the kidney, and inside the kidney it can be turned into urobilin, and that's yellow. And when you urinate, that's what contributes to your urine being yellow. And so if you have high concentration of urobilin, you're going to have more of a dark yellow pee. If you're hydrated, you're going to have more water, and it's going to kind of dilute it, and it's going to be more of a light color. But yeah, urobilinogen can be taken to the kidney, and it's a yellow in color, and that's what makes our urine yellow. Okay, and then kind of the last thing we'll do, urobilinogen, it can do neither of these two routes, and it can just keep going down the intestines, and it's going to be called sterobilinin is the name, and it's brown in color, and that's what contributes to our feces being brown. So here in the kidney, got turned into urobilin. This yellow here, sterobilin is brown. But that's like the breakdown of red blood cells. Kind of just go over it again. They're inside the blood after 128 days. A macrophage comes, gobbles it up, breaks it down. The globin, or the polypeptide chain, gets back into like the amino acid, and gets them back to use like the recycling center, which I know they're made in the bone marrow, but I think I read something about the liver too, so I don't know if it's stored there or can also go there, but we'll just focus on bone marrow for now. So that's where the globin can go. The heme gets broken up into the iron and the heme group. Both of those can either go back to the recycling center, or they can keep going. The iron can kind of make its way to be stored in the liver, and the heme can make its way, it's going to get turned into biliverdin, or from when it loses the iron, it's going to be turned into biliverdin. Quickly, it's going to be turned into bilirubin. And then bilirubin is going to enter the kidney. It's going to get its glucuronic acids. Now it's going to become conjugated bilirubin. And then from here, it can go to the bile. In the bile, it's going to lose the glucuronic acids, and it's going to become urobilinogen, where it can either go back to the liver in the enterohepatic cycle or enterohepatic urobilinogen cycle, or the urobilinogen can go up to the kidney where it can be turned into urobilin, which is yellow, or it can just keep on doing neither of those, keep going and be sterobilinin, which is brown, and be pooped out. So another thing I want to mention with this is jaundice. Jaundice is a disease that's found a lot in newborns, and that's due to their liver not being completely matured. I'm not 100% confident in this part, but from my understanding, if you have a buildup of bilirubin, I think that might be because you have like a lot of red blood cells being kind of broken down and destroyed, and that can lead to jaundice. And jaundice can cause like a yellowish color in the eyes and in the skin, and that's because I think bilirubin is yellow, and that's what contributes to that. And it's also, you can see bilirubin can be called unconjugated bilirubin because it's not conjugated yet with the glucuronic acids. But also it can go into the bile, and I'm trying to remember, but I think for some reason, it can go into the liver, and for some reason in the liver, I don't know if it's because these glucuronic acids don't attach, that's probably what it is, but for some reason the liver's not functioning properly like we talked about in newborns because it's not matured yet, and it's just going to stay as bilirubin or unconjugated bilirubin. And in that case, since it's not being changed into the conjugated form and going through the cycle, you can just have an excess of it, and that's what causes jaundice, or it's not being excreted as much as it should. So you just have a buildup of it. That's kind of from my understanding, a buildup of that, and that's what causes jaundice. So that's Unit 3, Question 3. That's Part 1. I'm going to do the other part here in a little bit once I study a little bit more, but be back in a minute. Okay, this is one of the last parts of the essay question for number 3 for Unit 3. We're going to talk about hemostasis, and to break down the word, hemo is blood, and stasis is standing still or slowing like the flow of a fluid. So for this, it's blood stoppage pretty much, or the stoppage of blood, stoppage of the flow of blood. And that's when we get a cut, or one of our blood vessels gets not ruptured, but gets damaged, and there needs to be something to stop or plug that damage within the blood vessel. So we're just going to talk about these first two right now. There's 3 parts of hemostasis. I'm not going to get to the third one right now because there's a class that's going to be coming to this classroom in a little bit. But these first 2 I'll talk about. So for vascular spasm, that's the first thing that a blood vessel will do. And it's pretty simple. It's a reflex. And if the blood vessel is damaged, the reflex is to vasoconstrict. And that's number 1, going to decrease the flow of blood going through that blood vessel. And so that's going to decrease the amount of blood that can leak out or go through that opening. But also what it can do, and sometimes it can contract enough where it will completely shut off, where it will completely plug that hole. Because if the blood vessel is lengthened out, that hole is going to be bigger. But if it constricts down, it's going to shrink together, and sometimes it can completely plug that hole. But with that, although it is effective, it's temporary. It's not something that is long-lasting, and so it's very helpful, but it is a temporary solution. So that's why we have these other 2. The platelet plug and the clotting cascade, those both are pretty much meshed together. They work together to fill the hole or fix the problem or fix the damage within the blood vessel. For the platelet plug, we're going to talk about it first. So right here we have a blood vessel, and up here we have the collagen or the basement membrane, and beyond that we would have smooth muscle. but in this case I didn't draw it. But yeah, for vascular spasm, we have the smooth muscle right here. See, I don't know if you can tell, but there's a hole within the vessel. And if we vasoconstricted due to the reflex, it would shorten up. And as you can see, that hole would either be shorter or it could be completely closed due to the shortening or the constriction of the blood vessel. But like we talked about, that's only temporary. So the platelet plug, let's talk about what that does. So we have the blood vessel. These little bumps right here are endothelial cells. Those are very important. So let's start. We get a hole or we get a damaged part in the blood vessel. There's exposed collagen. And when that happens, the endothelial cells nearby are going to release something called von Willebrand-Something's factor, VWF. And those are going to release and be excreted, and those are going to make, they're attracted to negative charges, and they're going to make their way over to the collagen, and they're going to bind to the collagen. And those are the anchor from the collagen to the platelets. So let's say the VWF is released. It's going to bind to the collagen, and then we have a platelet right here, and on the platelet you're going to have a VW receptor, and that's going to be attracted over here to this VWF on the collagen, and it's going to bind. Once that binds, once that VWF receptor is bound to a VWF, it's going to activate the platelet, and it's going to go through a transformational change. And with that transformational change, it's also going to, within the platelet, you have these vessels. There's alpha and dense vesicles, and they both release stuff, but what we really care about, at least for what we talked about in class, was dense vesicles, and those are going to release ADP. And ADP, why that's important, is ADP is going to go and it's going to activate other platelet cells. So it's going to be released, and here's a platelet cell. It gets activated by the ADP, and it's going to go through this transformational change. And then it's also, because it's activated, it's going to release from its dense vesicle ADP as well, and that's going to spread to other ones, and it's like that's what they call it, the cascade. Because one gets released, and what it's released goes to, let's say, 2 more, and those 2 more, since they're activated, they're going to release, and those 2 that release ADP, that's going to go activate 2 other ones, so it's a big cascade, and it's very effective. And so another thing we have is the fibrinogen, or fibrinogen, and I'm trying to remember where that comes from. Let me look it up real quick. So this is saying the fibrinogen is released from the platelets themselves. I'll have to check myself on that, but yeah, the fibrinogen is released from the platelets, and then on the platelets they have fibrinogen receptors, and so the fibrinogen, they are what anchor or connect platelets to other platelets. And so we talked about the VWF, that's what anchors the platelets to the collagen, but the fibrinogen are what anchor or connect platelets to other platelets. So as you can see right here, these fibrinogen with their receptors on the platelets are linking them together, and they're kind of building a team. Like if you had a bunch of people just grab and hold arms together, that would be the fibrinogen holding together, and you can get kind of a net-like formation. But yeah, so that's gonna plug, and pretty much with what's gonna happen here, you're just gonna get a big buildup of all these fibrinogen. And in the blood vessel, they're not gonna really come out this far. This is like just drawn out bigger, so you can see a little better. But pretty much you get the point. you're gonna get a plug from all these platelets that are activated, and they're gonna be connected to other platelets through fibrinogen. And once they're activated, they're gonna release more chemicals from their vesicles, from the dense bodies, that's ADP, and that's gonna go and activate other ones. They're gonna do a lot of recruiting and finally build up a nice little plug that covers this damaged area, and it's gonna plug it up. We call it the platelet plug. And that's about all I want to talk about with that. The clotting cascade, that goes right along with this, but I'm not gonna have time to cover it right now. I'll do that in a little bit. But a couple things I want to mention here is a thrombus versus embolus. And so a thrombus is this plug right here. If this plug were to break off and start floating down the blood vessel, that would be a embolus. And that's what can lead to like a pulmonary embolism. An embolism is when this embolus or whatever makes its way. And yeah, it's gonna be fine for a while because it's in a big blood vessel, but eventually it's gonna make its way to a smaller blood vessel and it's going to clog. And that's gonna stop the blood flow and lead to oxygen not being able to be delivered to the cells. Something important, platelet regulation, is we don't want this happening all the time because it's gonna lead to unwanted clogging or unwanted connecting of platelets. And so we need something to regulate that, and just, when it's not time, we don't want it going on, but when it's time, we want it going on. And so the platelet regulation, that's what's gonna kind of manage or regulate the formation of platelet plugs and the connections of those. And so platelet regulation, calcium is a huge player in that because it aids to the granular release, and it acts as a messenger for the platelet activation. What I think it does, I'm not 100% sure, but if you remember, we talked about calcium and how with neurons, it would aid in pulling the vesicles to the end of the membrane to release the chemicals. It allowed those, can't remember what they were called, kind of those arms on the membrane, arms on the vesicle. Calcium would come and allow them to grab each other and allow the vesicle to be pulled to the membrane to release the whatever chemical or neurotransmitter. That's kind of the same thing here, I think, because these are granulars or vesicles, I guess you could say, and maybe the calcium assists in that way, I'm not sure. But calcium is important, and that's what aids in the granular release of the ADP and the other ones. And so first, one thing that it can do, one mechanism, is it can block the release of calcium. Another one, it can activate this signaling molecule, prostacyclin, and that's going to activate the G protein, which is going to increase CAMP, and increased CAMP is going to increase the activation or the work of a pump that removes calcium from the platelet. Because I probably should have mentioned this, because calcium in the platelet is what's aiding in the release of the granulars, and when the granulars release ADP, they activate more. And so if we remove calcium from the platelet, there's going to be less calcium to help aid the granular release, which will then lead to less granular release, which will lead to less release of ADP and those other chemicals that activate other ones. So if prostacyclin increases a G protein that increases CAMP, and CAMP increases a pump that removes calcium, there's going to be less calcium. It's going to decrease that activation of other platelets, platelet regulation. Mechanism 3, ADPase, is going to chop ADP into AMP. And AMP, reason why that's important is AMP is what goes and activates other ones. If AMP is being turned into AMP, it's not going to be able to do that, so there's less ADP to go around to activate other things. Because also kind of what is cool, ADP, the reason why when it is activating, what it does, it goes and inhibits, ADP on the cell goes and inhibits prostacyclin. And if prostacyclin's inhibited, there's not going to be the release of CAMP through that G protein. The pump's not going to be working as much, which is going to keep more calcium inside of the platelet, which is going to allow for more release of the granulars. So that's how these two kind of tie together. But again, if we take out ADP, it's not inhibiting prostaglandin. Prostaglandin is going to signal the G protein to release CAMP. CAMP's released to increase the pump to pump more calcium out, which is going to decrease the granulars being aided in release. This one, I wrote it down, but I'm trying to remember how. Okay, NO blocks the release of calcium. Let me see how this works. So this is saying NO has a crucial role because it acts as a vasodilator and inhibitor of platelet aggregation. Let's see. So it promotes the relaxation of smooth muscle, leads to vasodilation, which helps maintains proper blood flow and prevents excessive platelet contact with the vessel wall. NO directly inhibits activation and aggregation of platelets. It does this by increasing levels of cyclic GMP, which reduces platelet activity pathways. So somehow, I don't know, it decreases the intracellular calcium concentration. I'll have to look into that a little bit more, but that's how these two bottom ones play a part. But somehow NO also helps with platelet regulation. Yeah, that's those two. Again, I'm gonna talk about this one more in a little bit, but these two are tied together and are happening at the same time. All right, so I had this in a classroom, but then class started, so I wasn't able to finish. So this is the part. So this is the clotting cascade or coagulation cascade. And important things about this, there are several pathways that lead to this cascade, or that lead to the clotting. And one of them is the intrinsic, which is right here. I don't know if you can see my little black dot. Maybe I can change it to a different color. Let's see. Okay, that red right there. So the intrinsic. The other one is the extrinsic, because that happens outside of the blood vessel. Intrinsic happens inside the blood vessel. And then we have the common pathway, where both of them eventually converge into, and that's factor, that's number 10. But to start out with this, what I want to start with is the role of fibrogen and when it turns into fibrin. And so if I zoom in a little bit, I'm not loving this. Let me see if I go to this picture, if it's more clear. Yeah, this looks like it's maybe more clear. So maybe I'll just do it on this, at least for right now. So with both of the pathways, they eventually lead down to, I'll go more into them in a second, but they eventually lead into the common pathway, which is active number 5. It goes from inactive number 5 to active number 5. And then it goes down to, let me see if I can write on this as well. Okay, here we are. Oops. So we go from inactive number 5 to active number 5, and I'm just gonna point with this right here. Hopefully you can see. But yeah, and then it goes to, then active number 5 is able to activate number 2, which goes from prothrombin to thrombin, or 2 in the active state. And thrombin, what it does, it activates fibrinogen, which is inactive, kind of floating in the blood, and then it turns into active 1, or the fibrin strand. And so we kind of talked about fibrinogen. When it's inactive, it's right here. It's kind of just floating within the blood, and it's soluble within the blood. But when thrombin comes, it activates it, and it turns it into more of a strand. It connects them, and it becomes insoluble. And all these strands of fibrin can be connective, and it makes a net-like structure. And I'm going to go back to this, and it makes a net-like structure on the plug. Because we talked about the platelet plug, but in order, like fibrin, its job is to kind of hunker down, kind of anchor down this platelet plug. And so the strands, as you can see, there's one right here that's going horizontal, and these other ones that are going vertical. Right now I'm kind of drawing these horizontal ones, but then they cross-link and create this net-like structure that holds down the platelet plug. And the reason why this is important is because it keeps the, let's see what I want to click on. It keeps the platelet plug from breaking off and moving down the bloodstream and causing trouble, like causing like a embolus. And but let's talk about the pathways a little bit. So the intrinsic pathway, it's independent of itself. And what it can do is within the blood, you have factor 12, and that interacts with collagen, and collagen will activate it. So when there's bare collagen, it will be activated, and it will become activated 12. And activated 12 will interact with activated, I think that's 11, and activated 11 will become, or inactivated 11 will become activated. And then that will activate 9, which will become 9, which is activated. And then it will activate 10, which will become active, inactivated 10 will become 10, activated 10. And once we have the 10 activation, that's when it becomes the common pathway. And the reason why it's the common pathway is because if you look over here with the extrinsic pathway, what happens is, so this is extravascular tissue, and if this is also damaged, what will happen is the extravascular tissue is damaged, as you can see right there, and it will release tissue factor, or number 3. And number 3, that activates factor 7 to its active form, and factor 7 can pretty much just go straight to 10 and activate number 10, which will then lead down here to the common pathway. So that's why it's the common pathway, because they both lead to this point, and it's the common point it leads to, it converges into. I also heard too that number 7 can also activate number 9. But I feel like the more important one, or at least that I was like kind of emphasized in the book and some of the videos I watched, was it does number 10, which is the active or the common pathway. But yeah, number 10, when it's active, I kind of mentioned it, prothrombin, or number 2, comes, is activated, becomes thrombin, and then thrombin can go and activate fibrinogen, which is soluble, just floating along when it's activated. It's able to bind together and create kind of a more gel-like strand, and that kind of interlaces, overlaps, and becomes a net over here to hold down the plug, the platelet plug. Let's see. Ninja Nerd. A few of these things in my video are from Ninja Nerd. This is one of them, kind of a cheat sheet, not cheat sheet, but just kind of easy way to remember it, because there's just kind of all those crazy factor numbers, but this is an easy way to remember it. So you start out with X marks the spot, that's 10, that's the beginning of the common pathway. But we know the extrinsic pathway, the extrinsic pathway and the intrinsic pathway both lead to the common pathway. So we'll start with the intrinsic pathway, which is 12, 11, 9, 10. So we pretty much count down from 12, 11, skip 10, 9, and then we're back on the common, which is 10. The extrinsic pathway, it's a lot shorter. It's quicker response. We start out with 3, or the tissue factor, and then we go to 7, which then activates 10, or the common pathway. The way to remember this one is 3, which is the tissue factor. 3 plus 7 equals 10. So remember, we have 3 and 7 over here. Here, just counting down from 10 or 12 to 9, but you're skipping 10 because 10's down here, leading to X marks the spot, or the common pathway. And then for going down, in the Ninja Nerd video I watched, it goes from 10 to 5 to 2 to 1. In the book that we have, we just went from 2 to 1, but with Ninja Nerd, he says you go to 5 to 2 to 1, so you do 5 times 2 is 10, times 1 is 10. So that's kind of a way to remember that. But remember, 2 is thrombonin. When it's activated, it starts out as prothrombin, but turns into thrombonin. And then when it's activated, it's able to activate the fibrinogen, which then activates the fibrin strand, or it into the fibrin strand. Just one sec. Okay, thanks for that break. Couple things. Let's see here. Sorry, one second. Okay, so yeah, that's the intrinsic, extrinsic pathway leads to the common pathway, which brings us to thrombin and the fibrin strand. Where do I want to go next? I guess there's also something called clot retraction. And the reason why that's important, I'll zoom in right here. And within so we have the platelet plug down in here, then we have the kind of net-like thing from the fibrin on top. But with the platelets, those have myosin and actin. And so they will contract together and shorten, like how myosin and actin shorten in a muscle. And this will end up, I think I want to draw this. Let's do blue. This will end up bringing this side of the endothelial cell and this side, or layer, I guess you call it. It brings it together because as those myosin and actin do their thing, they shorten, and we'll close this gap. And so instead of having it that wide, it'll kind of be more right here, and it can contract, and as it contracts, it brings it closer together. And also at the same time, there's different chemicals that are released that aid in kind of healing this layer and eventually bringing it back together. There's also different things that can be released too that can help proliferate the extravascular tissue. But just know from the platelets, I don't know if I was saying plasma before, but I meant platelets. From the platelets, There's chemicals that are released that will aid in rebuilding or proliferating the endothelial layer in those cells. Another important thing too is fibrinolysis. That's how you say it. And an important player in that is TPA, which is tissue plasminogen activator. And what this does is we don't want this clot in the blood vessel forever. Once this heals, we don't need the clot anymore, so we want to get rid of it. Fibrinolysis is what breaks down and gets rid of that clot. And so TPA is a key player. He's on the endothelial cells, I believe. And what he does, in the presence of fibrin, TPA will become more active. And when it's more active, we have plasminogen, which I believe comes from the liver, and it's inactive within the blood vessel. But as it's floating along, if we have TPA more active because of fibrin, it will take plasminogen and convert it into something called plasmin. And plasmin is what plasminogen is in its active state, and that will come and it will do its work on fibrin, and it will break it down, and it will get rid of it. And the fibrin will go back into, because we have fibrin over here, it will go back into its fibrinogen and just become soluble and kind of be recycled. But yeah, we don't want that blood clot floating around. We don't want it there forever. And so plasmin's super important to break that down and get rid of that blood clot. Another important thing is, let's talk about vitamin K for a second. Vitamin K is important because it's used for making clotting factors. It's an important vitamin for that. And there's a drug called warfarin or Coumadin, and that inhibits the recycling of vitamin K. So it reduces vitamin K, it inhibits it from being reused to make the clotting factors. So this will cause your blood to be able to clot less. And then another one is heparin. Heparin is a drug that's used kind of more for the intrinsic. This is more for the extrinsic. Heparin is a medicine that's been given for the intrinsic pathway, and that will also decrease the ability for the blood to clot through the intrinsic pathway. But there's tests that can be done. They're blood tests. One's the PTT, one's the PT. The PT is for the intrinsic and the common pathway, and again, that's for heparin. And the reason why this is important, if someone's on a medication, for heparin, we're trying to regulate the ability that their blood will coagulate or clot. And so we want to keep them within a healthy range. And so we want to give them the right amount or the correct dose of heparin. And so this test will tell us kind of the time it takes for the blood to clot. And so if the blood's clotting super fast, we know we want to give them more heparin. If we are doing the test and it takes a long time for the blood to clot, then we know we're giving them too much heparin and we want to reduce the dose. And so for this therapy, we just want to do it within a healthy range. So this blood test will help us. So if we're doing heparin, if someone's on a heparin medication, you do the PTT test, which is the intrinsic and common pathway. You wouldn't do the PT test if you're giving someone heparin. But if someone's on warfarin or Coumadin, you would do the PT test. And Coumadin, remember, is correlated with extrinsic pathway. And so kind of works the same as heparin. We're going to regulate or try to manage their blood coagulation or blood clotting. And so if we're giving them this medicine, their blood's going to clot less. But we want to do it in a way where we're going to do it within the normal range. And so we're going to do the PT test. And for the PT, if it's taking a long time to clot, we know we probably have given them too much, and so we want to down the dose. But if their blood's clotting super quick, we know we can give them more and just find that healthy range of where we want their blood to clot. To remember this, because you want to remember that PTT has to do with the intrinsic and common pathway, which is with heparin, because I believe heparin is released from the endothelial. We want to remember that PT has to do with the extrinsic with warfarin. So PT, TT, two T's together, they're hugging, they're connected. It's kind of intimate, kind of intrinsic. They're common. So remember the two T's is intimate, they're one, they're common, they're intrinsic, and that's with heparin. There's a lone T for PT, extrinsic, extrovert, wants to be alone, war, causes loneliness, warfarin. Kind of a way to remember that. But yeah, again, I got a decent amount of this stuff from Ninja Nerd. Don't want to take credit for that. But yeah, this is the clotting cascade or the coagulation cascade. And again, we have the intrinsic and the extrinsic, which lead into the common pathway, which eventually brings us to the fibrin strand, which gives us that net to bind it down. But then we don't want that net forever, so we have fibrinolysis, which can, we use TPA, which is a plasminogen activator, to turn it into plasmin, where plasmin comes in and breaks it down in a healthy, good way. And yeah, that's that. That's for Unit 3, essay question 3, the last part, so we're finally finished. And that's all I got for you.